In this video, we will discuss about antifungal drugs. These are fungicidal or fungistatic. Fungicidals are the drugs which destroy the parasitic fungi, and the fungistatic prevent the growth and multiplication of the fungi. Let us look at the characteristic features of the fungi. These are eukaryotic cells, and their nutrition is either saprophytic or parasitic. Fungi exist either in unicellular or multicellular form. Unicellular form of the fungi is called as yeast, and it is examples such as Candida and Cryptococcus. And examples for the multicellular is Aspergillus and Microsporum. And several types of fungi can assume either of the two form depending upon the environmental conditions. And examples for such type of fungi are Histoplasma and Sporothrix. And the fungal cell wall is composed primarily of the polysaccharides, and the fungal cell membrane is the lipid bilayer, and principal lipid is the ergosterol. And the cell membrane contains the enzymes necessary for the natural or normal function. Now let us look at the disease caused by the fungi. These are classified as superficial infections, subcutaneous or regional lesions, and systemic fungal infections. In superficial infections, it may be dermatomycosis or superficial candidiasis. Dermatomycosis is the infection of skin, hair, and nails. It is most commonly caused by the trichophytons and microsporum, and giving rise to the various types of ringworms or tinea. And in case of superficial candidiasis, it is the yeast-like organism may infect the mucous membrane of the mouth or vagina or skin. And in case of subcutaneous or regional lesion, such as zygomycosis, phycomycosis, and sporotrichosis. And in case of systemic fungal infections, it is caused by histoplasmosis, leads to histoplasmosis, coccidiomycosis, and cryptococcosis. And the main important is the Aspergillus flavus, which causes aspergillosis. Now let us come to the classification of the antifungal agents. These are classified as antibiotics, antimetabolites, azoles, allyl amines, and topical agents. Antibiotics are again classified as polyenes and heterocyclic benzofurands. Examples for the polyenes antibiotics are amphotericin B and hamycin. And for heterocyclic benzofurands, the most important example is the greasephulvin. And example for the antimetabolite antifungal agents are flucytosine. And in the antibiotics of the antifungal agents are again classified apart from the polyenes and heterocyclic benzofurands. The another class is the echinocandins. And examples for these echinocandins are micafungin and caspofungin. And in case of azoles, these are classified as imidazoles and triazoles. Imidazoles are again classified as topical and systemic. Examples for the topical imidazoles are clotrimazole or econazole, and for systemic imidazoles are ketoconazole. And for triazoles are examples such as fluconazole and oriconazole and fosaconazole. And in case of allyl amines, the most important example is the terbinafine. And lastly, that is the topical agents such as benzoic acid and butanafine and sodium thiosulfate. Now let us look at the site of action of these antifungal drugs. Amphotericin B, that is the polyenes, act on the cell membrane, and grisofulvin, that is the example of heterocyclic benzofurands which act on the microtubules system of the fungal cell, and flucytosine, which is the example of antimetabolite which act on the DNA of the fungal cell, and azoles which inhibit the conversion of lanosterol to ergosterol. Ergosterol is the lipid which is present in the cell membrane. By inhibiting the formation of ergosterol, therefore it will disintegrate the cell membrane, leading to the cidal or static effect of the fungus. And echinocandins, these are act on the cell wall, and drugs like terbinafine act on the conversion of squalene to lanosterol, thereby inhibiting this conversion of squalene to lanosterol, thereby ultimately preventing the formation of the ergosterol. This is the site of action of these antifungal drugs. Now let us look at the video of the mechanism of action of these antifungal drugs.
In the cell membrane is a lipid bilayer with proteins embedded in it. Ergosterol is the major sterol synthesized by 14-alpha demethylase enzyme that provides stability to the membrane. Glucan synthase complex, located in the cell membrane, is responsible for synthesis of beta-1,3 glucan chains. Elongation and cross-linking of the glucan chains with chitin imparts toughness to the cell wall. The azoles have broad-spectrum antifungal activity. They impair ergosterol synthesis by inhibiting the fungal cytochrome P450-mediated enzyme 14-alpha demethylase, inhibiting demethylation of lanesterol and accumulation of methylsterols. Lack of ergosterol leads to disruption of fungal cell membrane. Amphotericin has a high affinity for ergosterol in the cell membrane. Several polyene molecules get inserted into the fungal cell membrane as aggregates after combining with ergosterol to form a micropore through which amino acids, potassium, and other water-soluble substances move out. Echinocandins inhibit the synthesis of beta-1,3 glucan, which is required for cross-linking between chitin and beta-1,3 glucan that gives toughness to the cell wall.
Thank you.